Greetings, everyone, and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled, Addressing the Challenges in Maintaining and Analyzing Biotherapeutic Glycosylation. I'm Jeff Pogaliskis, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Minimizing batch-to-batch variability in product profiles while maintaining in vivo safety and efficacy remains one of the biggest challenges for the biopharmaceutical market. As the market for biopharmaceuticals continues to grow, manufacturers are increasingly challenged to improve process efficiency and develop methods that streamline workflows. Mandates from regulatory agencies, such as the FDA and EMA, require biopharmaceutical manufacturers to demonstrate comparable glycan profiles of critical quality attributes such as heterogeneous glycosylation patterns, setting up a daunting standard for product analysis and consistency. Let's meet our speakers for this webinar, who will discuss the complexities of glycosylation, its implication in bioprocess development, and the tools enabling precise control, quantitation, and characterization of glycan profiles. Ryan Boniface is a scientist in cell biology bioproduction R&D at Thermo Fisher Scientific. Mr. Boniface will provide insight into the products and workflows that can assist investigators in the manufacture of biotherapeutic protein, specifically through the quantitation and profiling of glycan. Shahir Khan is a senior staff scientist of Pharma Analytics R&D in the Bioproduction Division at Thermo Fisher Scientific. Dr. Khan will inform us on the various modalities for resolving and analyzing therapeutic glycan. Before we get rolling into Ryan and Shahir's presentations, I want to encourage everyone to submit questions for our Q&A session at the end of the presentations. We'll try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All right, with all the particulars out of the way, let's get our webinar started. Gentlemen, the audience is listening.
Hello, everyone. My name is Ryan Boniface, and I would like to welcome you all to the Thermo Fisher Scientific webinar on addressing the challenges in maintaining and analyzing biotherapeutic glycosylation. As the world leader in serving science, Thermo Fisher Scientific is passionate about helping our customers and companies improve quality, increase productivity, and develop highly consistent processes. There are many ways that we can support customers. Today we will focus on products and workflows that assist in the manufacture of biotherapeutic proteins. We have heard from our customers that glycan variability and product consistency is a major concern. Throughout the entire workflow, regulatory agencies require the monitoring of product quality. The sheer diversity in glycoforms has presented both high resolution and high throughput analytical challenges. This has presented an industry challenge. How can we integrate solutions to control, quantitate, and characterize glycan profiles to reduce variability and increase product consistency for biotherapeutic production? Protein glycosylation is one face of product quality, but it can affect structural and functional characteristics of an expressed protein. This can have profound impacts on in vivo half-life, efficacy, ADCC activity, and clearance rates. The example of glycan structures on the bottom right demonstrates complicated structures that can contain many branching sites. This leads to problems in sequencing or determining the glycan structure. By comparison, DNA and proteins have linear structures, which are relatively easy to sequence. Again, in the image to the right, you can see the glycan structures located on the cell wall. It isn't hard to imagine that this is the first thing that anything outside the cell will see. So not surprisingly, glycan structures are used for recognition. These recognition patterns can be seen in serum proteins, such as IgG, and this can determine if the molecular structure is foreign or to determine how long the protein will stay in the bloodstream before it is cleared. There are widely published process parameters and media and feed components, such as pH, dissolved oxygen, temperature, ammonia, viability at harvest, perfusion versus fed batch, all of these things either together or in combination can have an impact on glycosylation profiles. What I want to stress, and what the take-home message is, cell growth and product titer will not matter if your product quality is not right. Glycosylation isn't just a concern when producing glycoproteins in cell culture. There are many medical conditions seen here that can have impacts and or be the result of genetic changes that affect glycan biosynthesis and metabolism. Protein function is tightly linked to the glycan structure. Glycosylation affects biological activity, pharmacokinetics, stability, and immunogenicity. Here are some examples of what effects the glycan structure can have on biotherapeutic efficacy. Serum half-life, CDC and ADCC immune responses can be increased or decreased based on the terminal sugar on the glycan structure. Let's take a look at the ability to target selectable and controllable glycan profiles in cell culture. Every lab is looking to improve production throughput while creating a controllable and predictable glycan profile. Predictable glycan profiles enable consistency from run to run. Proper characterization and specific targeting of glycosylation optimizes efficacy of the biotherapeutic. Gibco EfficientFeed supplements and GlycanTune total feeds make this possible without having to make a choice between productivity and quality. I think it's worth repeating, good growth and product titer don't matter if your product quality is not right. Here's a simple experiment. Studying the titration of glycan modulating components in a supplement. The addition of glycan modulating components were effective in changing the glycan profiles. However, this supplement left quite a bit to be desired. As you can see in the graph, titration led to incomplete shifts in glycan profiles from G0F to more G1F and G2F. You can see a nonlinear response across all the titrations. There was a very small margin of error. Just adding a 0.03x supplement resulted in a 30% decrease in G0F compared to control. This experiment was not in any way predictable and would likely lead to a lot of extra work. This would include multiple rounds of titration experiments to find a specific supplementation level. Because of this and these inconsistencies, we abandoned this additive approach in favor of developing a complete feeding supplement that incorporated glycosylation modulating components along with a brand new feeding strategy. Let me take a few minutes to dive into our new strategy. This new strategy involves transitioning from an EfficientFeed Plus product, either EfficientFeed A+, B+, or C+, to our GlycanTune product, again, either GlycanTune A+, B+, or C+. At the very top of this chart is the EfficientFeed Plus control. And what this shows is daily feeding of EfficientFeed Plus from days 4 through 15. And you can see this by the gray boxes. This consists of only EfficientFeed Plus, and this will result in minimal galactosylation, or in other words, a high percentage of G0F. At the very bottom of this chart is the GlycanTune Plus control. This is daily feeding of GlycanTune Plus from days 4 through 15, and this appears as red boxes from days 4 through 15. This consists of only GlycanTune Plus. This will result in maximum galactosylation or a high percentage of G1F and G2F with dramatically reduced percentage of G0F. And what you have in between those two examples between those two control points are what we call transition points. So for an example, let's take day 11 from this chart as example. Feeding with EfficientFeed Plus is started on day 4 and continues all the way up to day 10. Then on day 11, the EfficientFeed Plus product, that feed, is replaced with GlycanTune. And then GlycanTune is fed from days 11 all the way up until day 15. So feeding is started with an EfficientFeed Plus feed and then switched to GlycanTune Plus. And the timing of this switch, or the transition, will lead to predictable changes in glycan profiles. Transitions to GlycanTune earlier in culture will lead to increases in galactosylation, or an increase in G1F and G2F. To demonstrate this, here is one experiment using the previously described transition times with EfficientFeed C+ and GlycanTune C+. The control on the left is only EfficientFeed C+, which has a glycan profile that is primarily G0F. The control on the far right is only GlycanTune C+, which has a glycan profile that is shifted from G0F to G1F and G2F. Protein quality was improved by decreasing G0F by almost 45% and primarily shifting to G1F and G2F. This was all done without negative impact to both cell growth and product titer. But in between those two controls, you can see the effects of the timing of the transition from the EfficientFeed C+ to GlycanTune C+. So what's really most important here is that there is a predictable and linear response. It'd be easy to hit glycan profile targets in between days due to the predictability of the shifts in glycosylation. And this can minimize unnecessary steps in process development. As I said before, you can improve your glycosylation profile without any negative impacts to cell growth and titer. You are able to maintain titer production, indicated in the titer curves here, and maintain growth rates while simultaneously controlling your glycan profile. The use of GlycanTune plus our efficient feeds equal efficiency and consistency. When used in conjunction with efficient feeds, GlycanTune enables preferential shifting from G0F towards G1F and G2F. It's also important to point out some of the other benefits of the GlycanTune Plus products. GlycanTune Plus was designed to be as easy as possible to reconstitute. It's as simple as 6 steps. Add water, mix in the AGT, bring to the final desired volume, filter, and then use. No pHing steps required. It's as easy as just adding water. You can see that other feeds require a high number of steps and pHing adjustments that are both time-consuming, they can pose safety risk to technicians, and can increase complexity, which can introduce errors. GlycanTune Plus can also be super concentrated, just like our EfficientFeed Plus total feeds. GlycanTune A Plus and B Plus can be super concentrated to a 3x concentration, while GlycanTune C Plus can be super concentrated to a 2x. To super concentrate, just add less water. If you were to make a 10-liter GlycanTune Plus feed solution, a 1x would need 10 liters of final volume, whereas a 2x and a 3x super concentration would only need 5 liters and 3.3 liters respectively. This leads to maximization of bioreactor usage. As an example, for a 100-liter bioreactor experiment using a traditional 30% total feeding process, the starting volume of the bioreactor would be around 70 liters. This would leave room for the 30 liters of feeding. If a super concentrated 3x GlycanTune Plus or an EfficientFeed Plus feed were to be used, That 30 liters is now decreased to only 10 liters, leaving room for roughly 20 liters of culture. So what you have is the ability to maximize your working volume. Now you can start with a higher initial culture volume of your bioreactor. You can increase your original starting volume up to about almost 90 liters, and then feed in that remaining 10 to 12 liters. So this right here is one of my favorite slides. What makes GlycanTune Plus and that transition process I spoke about such an important and powerful tool is that with just one experiment, using that transition timing that I showed earlier, using EfficientFeed C+, GlycanTune C+, and process development to transition to GlycanTune Plus, we were able to match the glycan profiles of 4 commercially relevant biotherapeutic profiles that we have measured. You can see that each glycan profile is within just a few % of the target biotherapeutic profile. This is critical for our customers who need to target very specific glycosylation profiles. So to review, GlycanTune Plus offers a number of advantages. There's a very simple dial-in process to tune glycan profiles. There's the ability to reduce G0F while increasing G1F and G2F without significant changes to performance. And all the benefits of the EfficientFeed Plus products that include simple reconstitution, just adding water, the ability to super concentrate to reduce feeding volumes, and to deliver unprecedented nutrient levels to achieve higher titers. GlycanTune is the only commercially available feed offering precision glycan control with increased titers in conjunction with the EfficientFeed Plus products. Now let's take a look at the glycoprotein purification. At the process scale, we offer thermoscientific capture select bioprocess resins and thermoscientific porous protein A and ion exchange resins. These products enable increased purity and yield and are designed to simplify research workflows and reduce time and cost of goods. Think about a standard downstream purification process. We typically train different unit operations together to move us from the cell culture stage to the purified product. Chromatography is heavily entrenched in these processes. Typically, there are 2 to 3 chromatography processes used. Today, we'll briefly focus on affinity options for capture, followed by ion exchange chromatography, which is commonly used in intermediate or final polishing steps. For capture select products feature unique, tunable affinity ligands that enable the affinity purification of any target biomolecule. Affinity resins provide high-fold purification in a single step. Poros offers high throughput, high capacity, base-stable protein A resin for the affinity purification of common monoclonal antibodies. We offer multiple complementary and novel affinity solutions that can greatly facilitate the purification of any biomolecule, including complex glycoproteins. Ion exchange chromatography is a common polish method for glycoproteins. Poros XQ is a high-capacity, salt-tolerant, high-resolution, strong anion exchange resin, featuring a fully quaternized amine surface chemistry. It can be used for the purification of recombinant proteins in bind and elute mode, or for monoclonal antibodies in flow-through mode. Poros XQ, when compared to 5 commercially available anion exchange resins, provides best-in-class capacity and salt tolerance, shown in the top graph. Poros XQ was compared to a variety of competitor resins, and the only resin that could provide similar binding capacity was CaptoQ. CaptoQ, however, does not provide the unique combined feature set of high capacity, high salt tolerance, and resolution. In particular, CaptoQ presents poor resolution capability. The lower left graph compares Poros XQ and CaptoQ resolving a 3-protein mix using identical conditions. Purification processes need to be efficient, which means that each unit operation needs to drive a purity gain. Therefore, a unit operating that simply provides capture concentration is not beneficial to the overall purification process. In the next graph, we have Capto Impress. And while it does have similar resolution, it does not have the binding capacity and salt tolerance as shown above. Poros XQ is the only resin that combines high resolution with high binding capacity in a wide range of salt concentrations. We offer novel affinity and ion exchange solutions with superior performance and established reputation for both purification of recombinant proteins and impurity removal. With robust quality, manufacturing, and global support, creating a simplified process with a reduced cost of goods for biopharmaceutical drug development.
Shahir Khan. Thank you, Ryan, for that very nice overview of the available tools for precision glycan control, increased titers, and glycoprotein purification. Following along the lines of what comes after an analytical scale purification, I will cover the products and the analytical tools used to analyze glycans. Before moving to technical aspect of this presentation, let's have a quick look at the regulatory requirements of glycan analysis. We know that more than 2/3 of recombinant biopharmaceuticals are glycosylated. Since the biotherapeutics offers a rich and promising frontier for development in biopharma industry for innovator and biosimilar molecules, FDA and EMA define glycosylation characterization requirements, and manufacturers are obligated to comply with these requirements. I'm not going to read the complete text here. Suffice is to say that since many biologicals have sugar chains that impact their safety and efficacy, FDA and its European counterpart EMA issued guidelines requiring that glycan structures should be characterized and attention should be paid to degree of glycosylation. As monoclonal antibodies come off patents, biosimilar race will heat up. In fact, it already started. In July 2014, FDA accepted the application for first biosimilar, and in March 2015 approved first biosimilar in USA. FDA and EMA have defined critical factors in determining the similarity to ensure glycosylation is well characterized in biotherapeutics and is highly similar to reference product. Because of complexity of glycans, their analysis is more challenging than any other biomolecule. There are a lot of technical challenges for glycan analysis. First of all, they are very complex and diversified structures, as you will see it in preceding slides. They have no chromophore or the fluorophore, and mostly they are not charged. Typical analytical methods used for glycan analysis include structural characterization by NMR and mass spectrometry, HPLC, normal phase, reverse phase, helix methods, and the capillary electrophoresis. And there are some hyphenated methods such as LCMS and CMS so on. To assist you in your glycan analysis, Thermo Fisher Scientific has developed a repertoire of tools and software to address challenges associated with glycan analysis. These tools were developed to improve sensitivity, reproducibility, selectivity, and overall ease of use as you utilize techniques such as CE, UHPLC, HPLC, LCMS for your glycan analysis. As you will see in this slide, that glycans are composed of monosaccharides, and they are typically 9 monosaccharides which are present in human glycan. And monosaccharides can be connected by an array of linkage stereochemistries and regiochemistries, leading to a large structural diversity, as we will see in this slide. So this slide shows the regiostereochemistry of the glycans. The sugars have several hydroxyl groups, and the second sugar can be attached at different places, and they also can be attached in alpha and beta configuration, which makes these molecules more complex. This slide shows the possibilities of isomers of oligosaccharides as compared to DNA and peptides. As we know, DNA peptides are straight chains, something like beads on a string, as compared to glycans, which can be branched. And just consider for a moment the example of a pentamer of a DNA or peptide. If you have the homopolymer of DNA and or peptide, you can attach them in only one possible way. If you have the heteromer of a DNA and peptide, they can be attached in about 120 possible ways. But when you move to glycans, the possibilities increase exponentially. Just for sake of example, let's consider the pentamer again. If you have the homopolymer of glycan, they can be about 18,000 different isomers. But if you happen to have the heteropolymer of a glycan, the possibilities goes to more than 2 million. But if we add one more sugar to it and we have the hexasaccharide heteromer, which is quite small in size, is about 100,000 molecular weight, but can have about 100 million possible isomers. However, in reality, this situation is simplified by nature, since glycans are synthesized by specific glycosyl transferases, and in many cases, only few isomers. Often, only one exists. Nevertheless, the structural determination is quite challenging for glycans. Current glycan analysis methods involve laborious multi-step sample preparation that takes anywhere from a day to multiple days for 96 samples, followed by single-channel LC or CE separation. Applied Biosystems Glycan Assure Glycan Analysis System is the first glycan analysis platform that combines both high throughput and high data quality. Furthermore, it offers an integrated glycan analysis platform that helps save labor, time, and cost of analysis. Glycan Assure system offers simple and easy workflow, which includes magnetic bead-based sample preparation, multiple fluorescent dyes for glycan labeling. We use industry standard APTS and our own proprietary teal and turquoise, which offers more sensitivity and selectivity. And our instruments are multi-capillary. There are two versions, 8 capillary, 3500, and 24 capillary, 3500 XL. And they are with the fluorescent detector. And the instrument comes with the assay-specific software. In comparison to traditional glycan analysis method, Glycan Assure system is able to process and finish CE analysis of 96 samples in one work day, which is 7 to 9 hours, and does not require any vacuum centrifugation or toxic sodium cyanoborohydride. The amount of hands-on time is less than 3 hours, while the data analysis takes less than 1 hour to perform the analysis of 96 samples. Let's have a look at the sample prep workflow. The sample prep for the glycan analysis is started with protein A purified sample. The first step is the deglycosylation using PNGSF enzyme. The incubation is done at 50 degrees Celsius for 1 hour, followed by glycan purification using magnetic beads. The next step is the dye labeling. As I said earlier, we can use the industry standard APTS. The reaction time is 2 hours at 50 degrees. Or we can use our proprietary dyes, teal or turquoise, which are faster reacting, and the reaction is complete in half an hour at 60 degrees. The next step is the dye removal, the excess dye removal, again using the magnetic beads, which takes about 30 minutes for 96 samples. The teal dye does not require any dye removal step. Now the samples are ready to be loaded on C instrument for analysis. And as I earlier said, complete sample prep process and the analysis of 96 samples can be completed in 7 to 9 hours with less than 3 hours of hands-on time. And the whole process is automatable. We also have the assay-specific software for fast data analysis and reporting. It takes less than 1 hour to perform the data analysis on 96 samples. There are several ways to visualize the data. You can see the relative abundance of your glycans in your samples, and you can also plot selected glycans from abundance plot to see the trends and quickly pick up the outliers if there are any. So it's very user-friendly data analysis software and has some really nice, cool features. Reproducibility. This slide shows the reproducibility of our system. The data was generated using 15 known glycans, which were labeled with APTS dye. They were mixed in unequal amounts and run on Applied Biosystems 3500 XL protein quality analyzer. 96 well plate loaded with sample was run 3 times, and electrophoregrams were overlaid. Presence of an internal standard in every sample enables perfect alignment across 288 injections. So what you see here is the overlay of chromatograms from 288 injections. Same experiment was repeated here with APTS labeled plasma glycans, and they were overlaid, and you can see the perfect alignment of the samples. In addition to Glycan Assure system, Thermo Fisher Scientific has a repertoire of other products to assist you with your glycoanalytics needs. These products are available to support the analytical workflows that enable you to generate the necessary data to characterize your biotherapeutic. The Vanquish Flex HPLC system provides a variety of mechanisms for separating your glycans. Separations are based on differences in size, charge, composition, and isomerism, and can be performed utilizing multiple mechanisms such as the reverse phase, ion exchange, hydrophobic interactions, and size exclusion. For LC-UV or fluorescence of intact glycoproteins, there are several options based on hydrophobic and ion exchange chromatography that provide fast, high-resolution highly selective separations optimized for monoclonal antibodies and other intact glycoproteins. In addition to providing separation, Vanquish also has 4 detector options which are currently available. We have dioxide detector with light pipe technology for ultimate sensitivity, providing 6x signal heights and 14x signal to noise ratio. We also have variable wavelength detector, perfect for routine analysis, fluorescence detector for labeled glycans, or even intact monoclonal antibodies which have natural fluorescence. And we also have the universal charge aerosol detector, providing valuable, which is proving valuable in the analysis of non-labeled glycans. As a result of 4 different detector options, the Vanquish provides to use sensitivity, linearity, and the flexibility to switch between detectors. For the release glycans, each designed to specifically meet your needs for selectivity and optimal separation. For neutral glycans, Aquapore 150 amide helic column is ideal. Shown here on the left is the performance of the column separating bovine fatty glycans designed for the separation of hydrophilic biomolecules in helic mode. This column has high retention of a broad range of hydrophilic analytes. In the middle, shown here is the separation system for quantitative, and structural characterization of uncharged and charged glycans. Glycan pack AXH1 LC columns provide simultaneous separation of glycans by charge, size, and polarity, allowing you to achieve high resolution and high throughput analysis with unique selectivity for biologically important glycans, either labeled or native by LC fluorescence and LC-MS spectrometry methods. Now moving towards the right, for industry-leading extra high resolution of labeled and native glycans, the new Glycan pack AXR1 is available. This column provides mixed mode separation using both anion exchange and reverse phase retention to provide separation based on combination of charge, isomer structure, and size. Shown here is the result of the same charge profile as in the middle chromatogram, with the exception that the peaks now are highly resolved within each charge state. And as you can see, the number of peak resolved are higher than the other 2 systems. If additional characterization and specificity is needed for your glycoprotein analysis, then LC-MS is the detector of choice. To identify as many intact glycoproteins as possible, as well as various glycoforms, a combination of high mass resolution, accurate mass, dynamic range, sensitivity, and acquisition speeds are required. And all these aspects are offered by Q Executive Series instruments. The complex branching and isomeric nature of glycan, as we have seen earlier, pose significant analytical challenge to the identification of these structures. In addition to characterization of the sugar sequence, the analysis must elucidate branching, linkages between monosaccharide units, and the location of possible sulfate or phosphate groups. To ensure comprehensive structure elucidation and structural isomer differentiation, a combination of high mass resolution, accurate mass, sensitivity, multi-stage fragmentation, and multiple fragmentation techniques are required. All these aspects are available on Orbitrap Fusion Lumos Tribrid MS system. It gives the flexibility in fragmentation. It offers the CID mode, collision-induced dissociation, high-energy collisional dissociation, electron transfer energy dissociation, and electron transfer higher energy dissociation. So there are several options for fragmentation available on this state-of-the-art instrument. The availability of multiple fragmentation techniques such as CID, SCD, ATD on this Orbitrap Fusion Lumos offers a level of versatility required for thorough glycan structural elucidation. When implying LCMS analysis, data-dependent CID or SCD can be used for structural elucidation, whereas low-energy CID predominantly generates glycosidic bond cleavages, where SCD can produce glycosidic as well as cross-ring fragmentation. HCD fragmentation mode provides necessary cross-ring fragmentation for glycan linkages and branching information. Furthermore, HTSCD affords the ability of near complete sequence coverage and confident glycosylation site localization relative to HCD or ETD method. These techniques support the glycopeptide analytic workflows for determining the location of the glycan within their respective glycoprotein. In summary, there were three main areas of discussion in today's webinar: growth and purification, which was covered by Ryan, and the analysis part, which I just discussed. In growth, we demonstrated the ability to control the glycosylation in cell culture. This enables an increase in protein titer without compromising the quality, resulting in productivity. For purification, we demonstrated the use of affinity and ion exchange solutions for glycoprotein purification. Finally, we demonstrated multiple ways to measure and identify the level of glycosylation with tools such as Glycan Assure system, which includes sample prep, multi-capillary C instrument, and dedicated software, and Vanquish Flex U HPLC system, Helix Glycan Pack columns, and our Orbitrap mass spectrometer. With such a wide range of solutions to offer, it can be challenging to find one that can best serve your needs. To facilitate this, we have created a selection tool which you can find on Thermo Fisher website. This is an interactive tool where you can quickly find the workflow of your interest. The tool then will show the products that support the workflow and put you in touch with an expert who will be happy to answer any question that you might have. We encourage you to visit the tool after the webinar. This is a highly valuable tool which can potentially help you in controlling, identifying, and measuring glycosylation. With that, we come to the end of the presentation. We thank you for your attention and attending today's webinar. Thank you.
Thank you, gentlemen. That was a great presentation. We provide our audience with some great information on how they can improve their targeting and analysis of glycan molecules and streamline their production workflows. So before we get started on the Q&A session, I want to let everyone know that this is their final chance to submit your questions to our speakers. So hurry up and send them in now. All right, so it looks like we have some really great questions that have already started to roll in. So bear with us for a few moments while we get everything straightened out on our side, and we'll begin the Q&A session.
Okay, so before we begin the Q&A, I want to introduce two additional folks from Thermo. Eric Niederkoffler is an R&D manager with Thermo Fisher Scientific. He oversees products related to Thermo's MESA brand. And Shelley Para is senior manager for global field applications team supporting customers using Thermo's porous resins and capture select affinity products. So why don't we begin answering the first question? And the first question is going to be for Shahir. The Glycan Assure looks great from an intra-assay perspective, but it is more important to understand inter-assay. How does the injection look on a multi-day test?
No, thank you very much. This is an excellent question. As a matter of fact, we have addressed this thing in our earlier webinar. Just to answer the question, yes, we have addressed this intra-assay issue, not only the day-to-day issue, but from instrument to instrument and capillary to capillary, and it looks great. We have very good, very tight CVs when we look from day-to-day variation, from instrument to instrument variation, and also capillary to capillary. And we also tested the instrument from across different sites at different locations, and the CVs were very, very tight. And I will suggest the audience to have a look at our earlier webinar, which is available on demand on Thermo Fisher website. Thank you.
Thank you, Shahir. And we have another question. How does Glycan Assure system work for complex biological glycoproteins, not just for IgG type of MAbs?
Okay, this again is a good question. We have looked different type of glycoproteins. In addition to monoclonal antibodies, we have looked highly silated glycoproteins like HPLC and ribonuclease B, and many, many other complex samples from our customers, and they look really good. The data we have produced, the customers really appreciated it because they have seen more peaks, what they have seen, and the detection was much, much higher. So our system is much more sensitive, and it resolves many more glycans which were not resolved in their system.
Thank you very much. And Shahir, Glycan Assure shows an FC glycosylation, but where would the ciliated glycans elute? How well does it separate monociliated to tetraciliated?
So I partially answered this thing earlier. So the silated glycans come first in our CE systems. The higher the negative charge, the faster they move. So for example, in the case of the fat chain, which is highly silated glycan, so we start seeing from the pentasilated ones, and then we see the tetra and the tri and the di and mono, and then at the end we see the neutral glycan.
And Ryan, we have a question for you. Are EfficientFeed and Glycan fed commercially available?
Yes. So our EfficientFeed and our EfficientFeed Plus products have been available for a number of years now. And the GlycanTune feeds have been available essentially from the beginning of this year. You can just go to thermofisher.com and either search for EfficientFeed Plus or GlycanTune, and you should see the list of products come up on the website.
And Ryan, the glycan data are very interesting. Have you tested the glycan tuning on more complex molecules? Were you able to increase glycosylation using this?
So internally, most of my work was on a more simple molecule, mostly IgG molecules. But there's no reason why this shouldn't work for more complicated molecules, EPOs, things like that that have more than 2 glycosylation sites. It should work in a similar manner. It should galactosylate, and at the same time, as you increase the number of galactosylation sites, you should also be able to open up more sites for sialation if the particular cell line of interest is sialating the protein.
And Share, a question for you. You mentioned that Vanquish has 4 detector options. Is there the option to have more than one of these options on a single instrument?
Good question. Yes, you can have more than one detector at one time. You can have all 4 of them. As a matter of fact, the instrument we have it in our lab, we have 3 detectors, which we use it all the time. So yes, it's possible you can have all 4 detectors at the same time and use them at the same time.
And Eric, we had a question come in for you. What flow rates are possible on the Vanquish system?
Oh, another good question. Yeah, the Vanquish X has a really nice wide range of flow rates it can use. It can go down to 1 microliter per minute all the way up to 5 milliliters per minute in 1 microliter per minute increments. And also it handles up to 1,500 bar as far as the pressure. So it's a really good, nice instrument. It gives great retention, reproducible retention times.
And Ryan, what do I do if I want to get less GO?
So if you were looking to get less G0, in the case with our media and our feeds, as shown in some of the graphs earlier on, I would suggest either a late transition to GlycanTune or maybe no transition to GlycanTune, just currently using either one of our EfficientFeed Plus products. But I realize that there's probably instances where customers are using their own media and feeds, and if it's a case where just the use of the customer's own media and feeds gets them to a G0 percentage that is lower than what they need to be, we can certainly engage our services groups, and we can then custom tailor a media or a feed to help them get to that point to either decrease G0 more or to increase G0 more. We have those abilities to customize anything.
And Ryan, can I use GlycanTune with my current feed?
So yes, there shouldn't be any reason why GlycanTune couldn't be used with a current feed. Certainly, I would recommend that the GlycanTune products are tried with the current feed that the customer is using. Since there are 3 GlycanTune feeds, an A+, a B+, and C+, I would recommend trying each one of those to see which one matches up best with the current feed in use. But if a customer is locked into a current feed and they really can't deviate from that, we can certainly, again, engage our services group, and we can incorporate our glycan enhancing technology into their current feed and work out a feed process parameters around that to help them out.
And Shahir, how is the GlycoAssure system's performance on highly silated glycans?
Oh, once again, it's a very good question. As I mentioned earlier while answering the other questions, the test article we use for the highly silated glycans in bovine fetching, which has from penta to monosylated glycans. And we have shown recently that they all can be resolved very well. As a matter of fact, we have a very good poster which will be presented in a glycobiology conference starting this week in New Orleans, where we have shown we can resolve all the glycans from penta to neutral glycans. And after the conference, that poster will be available at our website. So I would recommend the people who are attending the glycobiology conference just stop by and have a look at our poster, and later on have a look at the poster on our website. Very nice presentation of the separation of all classes of silated glycans present in bovine culture.
And Eric, someone says they noticed that the Axer-1 may be more highly resolved. However, it appears that the neutral glycans are not noted. Are they bound on the Axer-1 column?
Yeah, they are. So this AXR1 actually addresses both neutral and negatively charged glycans, and so the neutral come out earlier in the retention time. And And so some of the application notes, you actually will see that in the data shown. I think in this slide we didn't have that in that time frame, but it does come out earlier. They do retain on that.
Well, thank you. And unfortunately, it looks like we've run out of time, but please note that this webinar will be archived for 1 year on our website, www.genengnews.com. If you miss parts of it, you can watch it again, or you can recommend it to your colleagues and friends, which we highly recommend. Thanks again to the panel for the outstanding presentations, and thank you to our audience for your attention and for your thoughtful questions about various topics brought up during the webinar. I especially want to express our appreciation to Thermo Fisher Scientific, whose support made this webinar possible. And before the audience goes, we'd appreciate it if you would click on the green widget on the lower right-hand side of your screen to answer an audience satisfaction survey. We would be very grateful. Bye-bye for now.